Passive transport by me and Rachel. Membranes allow cells to carry out essential activities such as traffic regulation. The plasma membrane separates the internal components of a cell from its external environment. The framework is a bilayer of phospholipids, which are amphipathic molecules with hydrophobic interior regions and hydrophilic exterior regions. This attribute allows them to act as a selective barrier to materials entering and exiting the cell. Cellular membranes also contain carbohydrates attached to lipids and proteins. Cholesterol affects the fluidity of membranes by providing stability. Higher temperatures have less fluidity, and vice versa. Carbohydrates serve as recognition symbols for other cellular proteins. This allows them to play a role in determining blood types. Proteins act as enzymes in a membrane to break down polymers. One of the major functions of proteins is transport, which is defined as the movement of ions and molecules across a membrane. Substances can move across a membrane by diffusion and passive transport, which refers to high to low concentration, and facilitated diffusion, in which proteins provide a passageway, or even active transport, where molecules go against their concentration gradient. Osmosis. As you can see from the picture located at the bottom of the slide, hypotonic solutions have a low solute concentration compared to the inside of the cell, whereas hypertonic solutions have a high solute concentration. Isotonic solutions generally have identical solute concentrations on either side of the plasma membrane. Osmosis refers to the diffusion of water through a membrane from a solution that is hypotonic to hypertonic. The tendency of water to move into a cell causes osmotic lysis. Hypotonic solutions have more free water because they have a lower solute concentration. Since substances tend to move from an area of high concentration to an area of low concentration, free water will move into a cell. When a cell is placed in a hypertonic solution and too much water is released, it will shrink, a process known as crenation. All of this is explained because water is inclined to balance solute concentrations in order to achieve equilibrium. Okay. For the first experiment, we had 4 dialysis tubes labeled 1 through 4, each holding up to 10 milliliters of a solution. We first folded over and clipped the bottom of each tube to prevent leaking. We then opened the other end of each tube, which required a certain technique in which we placed the tubing under a stream of water and tried rubbing it open with dry fingers. But anyway, we then filled the tubes with a specific amount of sucrose and distilled water based on the 30% solution we were provided, which you can reference in the chart. Then we folded the open end over and fastened the dialysis clips. We then measured the weight of all the tubes before placing them into beakers of deionized water. We dried and measured the tubes 5 more times over the course of an hour at intervals of 10 minutes each. So here's a video of us preparing, I think, the tube for 6% sucrose. We're going to put a little first just to open it up. And then, yeah. And these are our 4 tubes all set and ready to go.
The first picture here shows what the dialysis tubes look like after the experiment. The section picture provides a close-up of the first dialysis tube. After weighing each dialysis tube at increments of 10 minutes for an hour, we had our results as displayed in these tables. So the first table shows the tube number, concentration, and initial weight, and the second table displays the 6 reweighings of all 4 test tubes in order. And as the tables show, the fourth test tube, which had a sucrose solution concentration of 30%, had a high weight change from 15.73 grams to 19.29 grams. And that means that the most substance is passively diffused in this dialysis tube. And we can also examine this in the following graphs. So here we have it for tube 1 as it changed over the amount of time. Then tube 2, tube 3, and tube 4. So after compiling this data, we can see that the first dialysis tube with a concentration of 0% sucrose had the lowest rate of diffusion, and the fourth dialysis tube with a 30% sucrose concentration had the highest rate of diffusion.